This is Dr. Wu. I'm making this video for Lab 3, Diffusion, Osmosis, and Dialysis. This is very interesting lab. First of all, we need to make sure those three concepts are clear. Diffusion is a process of a substance move from high concentration to the low concentration. Osmosis is a process that water molecule moves from high concentration to low concentration through semi-permeable membrane. There are three concepts under osmosis: isotonic solution, hypertonic solution, hypertonic solution. Osmosis is the process that water move from high concentration to the lower concentration through semi-permeable membrane. There are three concepts under osmosis. Isotonic means the solution has higher isotonic solution, means solution has an equal solute concentration in both sides. Hypertonic solution is the solution that has a lower solute concentration than that in another solution. Hypertonic solution is a solution that has higher solute concentration than that in another solution. Correspondingly, we have this two concepts used in blood cell. When blood cells are placed in high solute concentration solution, they form small clamps. This process is called cremation. However, if the blood cells are placed in a lower solute concentration, those cells begin to expand and maybe burst. This process is called hemolysis. The mechanism of the two processes are the same. They cause by osmosis. We will have a question about these two concepts. Third one is dialysis. The dialysis is the substance which is small molecules, including sugar, water, etc., can pass through semi-permeable membrane freely, but a large molecule like a starch cannot. So that's the concept of dialysis. In this lab, we'll use experiment to learn these three definitions, and in order to do that, three different tests are needed. First, the test for chlorine, use silver nitrate, and the solution will turn white. Test for starch, using iodine, the solution will turn blue. Test for glucose, using benedict, the solution will turn yellow. The purpose of this lab is to use dialysis to separate small size particle from a large size particle, and discuss hypertonic and hypertonic solution in a blood cell. I will demonstrate diffusion using these two solutions. One is just pure water, another is water plus agar. I add 1 drop of fruit dye in each solution. Then you see the size of each droplet in these two solutions. I will begin to time it. So the timer here shows for 4 minutes. After 4 minutes, the fruit dye diffused we can see the difference in diameter. So I will measure the diameter here. So it's about 5 centimeters equals 50 milliliters. Then this one is about 1 centimeters equals 10 milliliters. This is next experiment. I made a dialysis bag which is look like this. So the mixture of 1% starch, 10% glucose, 10% sodium chloride is inside is added inside this dialysis bag. Then I knot both ends. So I place this into a beaker with just the pure water by using the concept we learned, osmosis. We can explain the result. By using osmosis, we can explain the result we got. And the first result is this. These 3 tubes are for the test of chlorine. So the first one here is this solution. So this is the solution outside the dialysis bag at 0 minute. This is solution outside the dialysis bag at 40 minutes. And this is the content inside the dialysis bag at 40 minutes. So we see there's no reaction at 0 minute, and both has the reaction at 40 minutes. I record that result in a table on page 2. This one here is the same as the previous one. 0 minute outside dialysis bag, 40 minutes outside the dialysis bag, 40 minutes inside dialysis bag, the content of dialysis bag. So we see this test for starch, only the inside of dialysis bag has a reaction. Those two, there's no reaction. The color is from the ideal solution itself. This result has the same solution as the previous two. Outside the dialysis bag at 0 minutes, outside and this inside the dialysis bag at 40 minutes. And the test is for glucose. So we see these two has a reaction. The solution turn yellow. The blue means no reaction because the candidate solution is blue. So that indicates the presence of glucose in here and here. I record the data from the observation I just showing previously. So that's the reaction result for chlorine test. This is the result for starch test. This is result for glucose test. Then the time and with this information, you are able to answer the following questions. So in question A, I provide the diffusion distance, which is 50 milliliters in water and 10 milliliters in agar within 20 minutes. You can calculate the diffusion rate in these two solutions to see the difference. Then with the dialysis data, you can answer the question B and C. Then the last one, you just go through the information in the introduction part, you will find the difference between osmosis and dialysis. That's all for today's lab. Thank you for watching. Bye-bye.